well, we'll see what happens here. So, this is sensation and perception. I think we already established that, right? But let's talk about sort of our introduction to this, right? The first thing I want you to know is we are going to revisit a lot of topics in this class, right? We're going to come back around. Don't worry. This is probably, I'm just going to tell you right now, this is going to be the most difficult class you have taken in this department. Okay? I know it's a 300 level class. Those numbers are useless. A numbering system means nothing, so don't think it does. Okay? This is going to be a complicated class for you, not because I'm going to ask too much of you, because I'm not, believe me, but because the material is going to come at you in a way, and it's going to be new material that you've not heard of before, you've not thought about it this way before, and it's very abstract, right? And it's going to take us a little bit of time to grab on to those physical things that you know and that we have and put it somewhere that makes sense for you, okay? So this is definitely a class where you're going to be uncomfortable for a while, okay? And that's because I'm going to put nails in the seats next week. But you're going to be uncomfortable for a while with the material, okay? But I just want you to sit with that. That's fine. You have a great support system here. You've got me. Josh has taken this class, Marissa's taken this class. They both did really, I guess I can share that. They both did really well, or I wouldn't have selected them to be teaching assistants for this class. So you've got support if you're not grasping something, okay? That's fine. That's why we're doing the lab activities to reinforce what we talk about in the lecture. That's why you have this great book that's got all the additional resources. Take advantage of all of that. There are going to be things that don't make sense to you. They're not going to make sense to you today. But next Tuesday, when you're walking by the Cam Henderson Center, all of a sudden you're going to go, man, receptive fields, I got it. Okay? And it's going to hit you at weird times. And I promise you that will happen. Because like 3 weeks from now, somebody's going to come in and say, you know what, it actually happened right beside the Cam Henderson Center. Because that's how good I am at knowing these things. So hang in there. Don't get discouraged, right? If you're confused, if you're lost, come see one of us. We'll help you out. But just be ready. The other thing, we're going to keep, I like to think about this as a carousel, right? We're going to keep coming around, okay? A lot of folks teach this way, but they don't tell you they're teaching this way ahead of time, and you don't know what's going on, and you just get dizzy, right? I'm telling you ahead of time, we're going to go in circles, okay? We're going to come back around to something, and I'm going to constantly be saying, hey, remember that time I told you? And I'm going to tell you something that I've already told you once, and I'm going to say, look, this connects to that. And you're going to be building these structures, okay? So I'm going to let you know ahead of time. If you fall off, go get us a little bit of cotton candy and come right back. Okay, we'll pick you up. I promise it's going to be fine. There are going to be layers in this class. The book is written in layers. I already told you about those blue pages, right? Those are advanced pages. If you're excited about a chapter, read the blue page. If you're not excited, skip the blue pages, right? So we've got layers. I'm going to tell you things in class, okay? Sometimes things are going to be really exciting, sometimes they're not. If there are any musicians in the class, okay? Mason, you might get really excited when we start talking about the auditory system, because we're going to spend some time talking about sound waves and harmonics and things like that, right? Things that to musicians make a lot of sense. If anybody's an artist, you guys might get really excited when we talk about depth perception, right? If you do some 2D media of some sort, right? And so that stuff you might start thinking about this shape constancy and things, and that might get you really excited. You might not care anything about harmonics, right? But we'll come back and there will be something for everybody, okay? The other thing is I am definitely a reductionist, right? What does that mean? I really go down for that lowest level sort of explanation, right? And so in this class, we're going to really bring it down to the biological components, right? We're going to spend a lot of time talking about, if I'm talking to you about vision, first thing you got to know is about the eye, right? I can't tell you about visual processes if we're not talking about the eye. Before the eye, we've got to talk about light waves and light particles, right? So we're going to spend time talking about the physics of that particular stimulus so that you can really understand what we're dealing with here, okay? It's not going to be anything that's going to overwhelm you, right? But you just need to have some basic concepts here so we can move forward, and this will all make a lot of sense to you. All right, any questions about that? Anybody else want to leave? I mean, you've already done a lab activity. You might as well stay now, right? Maybe I should have led with that. As soon as you came in, I should just throw a bunch of lines at you and said, hey, here you go. Eat these, see what happens. I don't know. Mason, you had something?
I would have been okay with that.
You would have been okay with that? I thought so. You're a line kind of guy. I guess, should I tell him a pirate story?
Yes.
Yes? Eli likes pirates?
Yes.
Okay, that's cool. Do you know why they call pirates limeys?
They call pirates limeys?
Yeah. Okay, so often they'll call pirates limeys. Sometimes they'll refer to sailors as limeys as well, right? The reason for that is this great thing called scurvy. Anybody had that one? No, because your faces are all attached. Scurvy is a breakdown of the collagen in your body. Collagen is one of the #1 proteins that you've got that holds you together. If your collagen breaks down, stuff falls apart, right? Like literally, chunks of you will fall off. Okay. So what's the key ingredient in making collagen? Believe it or not, it's citric acid, right? So you need, or ascorbic acid. So you need this from citrus fruits, right? You need that acid. And so what they would do is they would pack like barrels of limes on these long distance voyages, right? Because we used to, if you were trying to sail across like the Atlantic, that took, somewhere between 3 months and the rest of your life. So it wasn't a real safe trip, right? It's not like now you hop on one of those Carnival cruises and you just, all those things are dangerous too, for different reasons. They'll tip over.
Leave you on them.
Huh?
Captains would leave you on them.
Yeah. I'm surprised they did. I mean, they left you on the ship?
No. I don't think it was Carnival Cruise, but there was this news. It's fairly old. But there
It
was
was
this
from like
one.
5 years ago. He
Yeah.
jumped ship and let half
Yeah,
the
the
people
captain
die.
Oh, I remember
Ship
that.
was
Yeah, that was like off the
beaching.
coast of Italy, right?
Yeah.
Yeah, I recall
They just
that.
now got the cruise
The ship
off
wasn't
there.
like in any
Really?
immediate
I've totally
danger,
given
but-
up on that story.
Yeah.
I was like, well,
I was just deep into YouTube one day,
yeah,
halfway through the documentary.
it happens. Don't worry about it.
And yeah, I don't think the ship was in any real immediate danger. It was just like beached or something, and the captain just up and left.
I always thought the captain went down with the ship.
That's the point.
That's the movies.
That's the movies.
This
Yeah,
is real life.
I know.
No. In the movies, musicians go down with the ship.
Well, there you go, Mason.
Don't look at me. I don't play the cello.
So, reductionist. For some reason, I feel like that's a Titanic joke, and I only saw 8.5 minutes of that movie.
I've never even seen the movie.
It's too long.
I've
That was
never
that was
seen
too long. The 8.5 minutes were too long. I should have only watched 4 minutes, and I would have been just as happy. So,
You know what
I'm
happens.
the ship goes down.
Exactly.
Okay, I didn't know if you knew that. Spoiler alert. Sank 100 years ago. All right, so there you go. That's important. Chapter 1, general principles. So why should we even study the senses, right? Well, it's a required class for some. No. There are reasons, right? Perception, and we'll talk about the difference between sensation and perception here in just a few moments. Perception, though, it seems really effortless and immediate, right? I mean, how many of you, when you open your eyes and you look at things, it just happens, right? You're like, oh, man, that's an apple. It's red. You don't even have to really think about it. You just know red apple and you're done. You just move on, right? We very rarely make mistakes in our perceptions, right? Now, there are some very key important times when people do make mistakes, right? And I'm certain like somebody's going to stand up and yell about this one time that the referee called something out when it was actually in and it made the difference, right? And you're like, that was in, that's important, right? And so but very rarely, right? You think about all of the other times that we make these very rapid, very fast perceptual judgments, and we're very, very good at that, right? How many times do you walk out the door successfully? Well, like almost every time. Very rarely do you walk into the doorpost, right? Because you're making that great perceptual judgment, and it happens. And it happens quickly, and most of the time we agree with other people, right? If we're all watching the same event, most of us are going to have the same perceptual experience, okay? Now there are some variations, we'll talk about that. What's really interesting about this is as quick and effortless as that is, it's in fact a very complex neural process, and it uses a major chunk of our brain, right? There is a massive chunk of your brain that is devoted to sensory systems, okay? Whether that's vision, audition, balance, vestibular system, touch, smell, taste, whatever it is, right? A chunk of your brain is devoted to that. And so if we really want to think about brain function, we want to learn something about brain function, well, we should really spend some time talking about sensory systems, right? Now, we do make misjudgments. Often these are in critical situations, especially if we're doing something hazardous, like driving, right, or in sports. Okay. There are outside factors, we'll think about this some, that will affect your perceptual assessments, right? So we really want to think about when you're in a stressful situation, when there are other competing things for your attentional resources, that we might make some mistakes, okay? All right, so basic definition. When we talk about sensation, we're talking about this very simple, straightforward stimulus and receptor interaction. Okay? Right now you don't really know what a receptor is, but you have specialized cells, nervous system cells, that are designed to receive, that's why it's a receptor, to receive some stimulus, whatever that is, right? In your eye, how many of you guys know about the retina? You guys know a retina exists, right? How many of you have ever been to an eye doctor? So somebody's looked at your retina, right? Like I'm glad to see most of you have been to an eye doctor at least once, right? Get your vision checked. Some of you, I think, go more often than once. I can tell that, right?
Don't mention.
Got things on your face, right? So I can kind of tell sign there. On the back of your eye, in the back there in the retina, you have photoreceptors, right? These are specialized cells in your nervous system that receive light. Okay? They're sensitive to light. When light hits them, they get excited and stuff happens. We'll talk about that process later, Lauren, so don't get too excited about it just yet. We're going to learn about it. Just hold on. I know you just want to take off there. That's the receptor. The stimulus is the light. It's that simple. If you walk outside and you smell a cheeseburger, it happens sometimes, right? We're not too far down from Fat Patties if the wind blows just right. If the wind blows just right, sometimes you smell industrial waste, and sometimes you smell a hamburger. So, kind of depends. Why do you smell that hamburger? Well, there are particles in the air of that hamburger, believe it or not. So there you go. You're already losing money when you buy a hamburger because particles are flying off that other people are smelling. Eat it fast. I know, right? Should come out wrapped in plastics.
To stand out by the vents where they, where all that cooking, where all the cooking gases get put out of.
I bet you could stand out there and instead of buying a hamburger, you could just smell enough of it to get full.
Yeah.
Is that why on Jimmy John's it says free cents?
I think so. They don't know what they're doing.
They
They don't know what they're doing. That's a bad business model. As I will tell you right now, it doesn't exactly work that way. It's not very efficient, but I assume it would work eventually. So there are particles of that hamburger that you're smelling. They're interacting with receptors in your olfactory system, kind of up in your nose, and that interaction causes you to, that's that sensation, right, that interaction. Now, perception goes a bit beyond that, right? And perception is like you saying, it smells like a good cheeseburger, right? That's your perception of that smell, okay? Your sort of emotional reaction to that, right? Anything sort of beyond that basic physical interaction between the stimulus and the receptor, that's going to be perception. Now sometimes, Mason, this is difficult to tell the difference between the two, right? It's kind of difficult to tell the difference between the two. Like, is this light coming in, it's hitting receptors, I'm perceiving that as red light, but you know, that's activating receptors that only are sensitive to that wavelength of light. Now, is that sensation, is that perception? That's a little fuzzy. And so I don't want you to spend a whole lot of time in that middle ground of sensation and perception, like what's the difference, but the things that are on the far ends, right? Those are pretty easy. Sensation, definitely. Stimulus, receptor interaction, that's it. Anything else is going to be perception. So we can kind of draw our definition that way. Does that make sense? Awesome. So there are different types of senses. Do not walk out of this class today and tell people you have 5 senses. Please don't do that. People will tell you you have 5 senses. That's ignoring 8 of them, right? There are so many more than 5 senses. So many things you're sensitive to, some things you don't realize you're sensitive to, right? You have receptors for that. We'll talk about those. Some things you do. When we talk about something called general senses, so we're going to group this, right? We group it into what we call modalities. Not a big deal. General senses, you have what are called somatic. This is tactile, so we think about touch, pressure, vibration, itch, and tickle. Why are we breaking those all down into different categories? They have different, exactly, Olivia. There's a different receptor for each of these sensations, for each of these, for this stimulus, right? And so, like touch and pressure and vibration, itch and tickle, things, different things cause those sensations and perceptions, right? So there's a different receptor for them. And so that's why we can break those down into different categories. We also have one for thermal, that's hot and cold, pain, and then something we call proprioception, right? Anybody in here nobody's in kinesiology, exercise science? Josh, it's not gonna help anybody but you. Proprioception is, and then there's another, like kinesthesis, if we really wanted to get into it. But basically these are like the awareness of your body position and the movement of your body, right? So how many of you can see your feet? Well, close your eyes, right, and your feet disappear. But you know where they are, right? You guys know where your legs and your feet are, even though you can't see them right now, right? You're aware of that. That's a sense, right? That's a sense people don't think about. We also have something called visceral senses. This is how you feel your guts. So if you stick your arm right inside, no, just kidding. Visceral senses are things that you're often not aware of, right? So you're often not consciously aware of your visceral senses. How many of you know what your blood pressure is right now? Nobody. How many of you know you have a blood pressure? Yeah, you got a blood pressure. Guess who knows your blood pressure? Your body does, right? Your body's monitoring your blood pressure. We have receptors that measure the pressure in your blood vessels, okay? And then your body's going to make some physiological changes to that. How many of you know the oxygen concentration in your blood? Nobody. Hopefully you can take a wild guess, right? None of you are gasping. I'm going to say it's somewhere between 99 and 100. Maybe 97 if you guys are slow breathers.
That's such a weird insult, you slow breather.
Yeah, well, you should be creative with your insults, right? Call people slow breathers. I don't know if that's, well, it is what it is. So, your body's aware of how much oxygen is in your blood, and you will adjust your respiration based on that, right? So that you can put more oxygen into your blood. Again, it's not something you're consciously aware of, but it's definitely a sense, right? It's a sense to know what's going on. The next set of senses, kind of a broader group that we have, are called special senses. We've got vision, we've got audition, we've got equilibrium, right? So these are vestibular senses. Are you upright? Are you inverted? Are you spinning in a circle? Okay. Olfaction is smell, and gustation is taste. It's kind of a nice little chart showing you
Can we get
the...
back real quick?
Yeah, I was
Just
about
want
to
to put those down.
Did you want me to stay back there when I went there? I mean, I just did this. Was that...
Stay. Perfect. Just a second.
That was an accident. I really... Now I don't know. It's not moving. I froze my computer. Yeah, no, it's frozen now. Great job.
Some may call it karma.
Is that glass right there?
Day one, we broke the computer.
So, two things here. One, you can get these slides online. In two different places, right? Yeah, it's totally broken. Man,
If you hit it harder, it'll go.
I think so, right?
Recursive maintenance.
Recursive maintenance.
Sounds like class is over to me.
No. No, we're gonna keep talking. I mean. See, there's always a trick.
Oh.
I know.
There you go.
Oh, I hit a bunch of buttons. Of course, the server's busy. So we group these into, group the senses into different categories, sort of based on different things. I'm not going to ask you a lot of questions about that, right? I think it's important to understand that there are broad groups. Those broad groups have differences. There are more than 5 senses, right? I think that's important. Document recovery, whatever. Yes.
Okay, I know that there are ways for you to like not be able to feel pain and like you can obviously lose like your vision and hearing and all. Is there a way to lose the feeling of like the P word that I can't say?
Proprioception, absolutely. And we'll talk about some interesting cases on that when we talk about body senses later. Yeah, there are folks who lose awareness of their body, right? And it's really weird because they'll like see an arm and they're like, oh, that's not my arm.
Like
Even
an
though it's
out-of-body
like
experience.
clearly attached to their shoulder, right? And they're like, that's not my arm. I can't even move it. Like I have to do this because they're not even able to send a signal out. Like they're not. It's weird.
I feel like I remember learning about something similar to that in cognition last semester.
Yeah, there's some weird stuff that happens. Kyle?
How does it work when like a body part falls asleep?
Yeah. Well, I mean, you're kind of losing a lot of that's like circulation issues, right? And so you're losing some problems there. You can also pinch like nerves. So yeah.
Does that not have like as much to do with this type of stuff as it does just the circulation?
Yeah, I mean, I wouldn't. I mean, you're not really doing anything with the receptors in that case. You know what I mean? Does that make sense? I mean, if you're
trying to though, and
yeah, but I mean, your receptors are still intact. They're fine.
right, but like
It's
the
just-
tingly feeling that you get, where's
Yeah.
like that coming from with the?
You know, I'm not entirely sure what's causing that. I think if I'm thinking about this from like an evolutionary perspective, I think the point of that is like don't do something that makes your arm fall asleep. Because that's a bad idea. Because if you do that long enough, you're gonna like completely cut off circulation and there's gonna be tissue damage.
Right, I'm just saying like falling
So
asleep
it's-
like that.
So I think it's like yeah. So don't fall asleep.
Just
That's
don't-
bad.
okay.
What about sleep paralysis?
Sleep paralysis is a whole separate thing. Don't worry
Like-
about it. We're not gonna talk about sleep paralysis in this class. You should sign up
It's
for my
crazy
440 class. There's an empty seat in there. We'll talk about sleep paralysis there. Yeah, this class, sleep paralysis doesn't matter. Did you get everything you need, Alex?
Yes.
Awesome. We should talk briefly about types of receptors. There are different ways we can categorize them. Structure is one way. So we have things called free nerve endings. That's these guys here. There's nothing on the end of those, right? There's nothing sort of covering them. This is really great for things like pain, temperature, tickle, and itch. Things we want to know about quickly without any sort of intervention, right? Without any sort of filter, without any sort of chance of reducing that stimulus. Do you know what I mean? So if you're feeling pain, guess what you should do? Whatever it takes to get away from that pain, right? I mean, that's how this works. Okay, so when you feel pain, you need to make a response quickly, because if not, there's going to be continued tissue damage. Temperature is also something that you want to feel quickly. You don't want to put your hand on the stove for 10 minutes to figure out if it's hot, because that's going to cause a lot of tissue damage. Okay, same thing if it's something cold. Tickle and itch might be things you don't really think about, like why would we want to know if we're being tickled or itched quickly. There's a really interesting thing to think about, and it's parasites, right? Like those little bugs that crawl all over your body and make you itch, right? Like lice and bed bugs, and ticks and spiders. And what are the other things, Marissa, that just
I
like...
don't know, please stop.
You don't know. Yeah, and they'll just, I mean, they'll crawl up your back, up your legs, I mean, they'll get in your hair, crawl around your ears, they'll give you all kinds of problems. You really gotta, it's tough business, right? It's not such a problem now because we have things like, well, we have showers. Those are awesome. We have clothes, which, I mean, some people might argue not as awesome, but they're still awesome, trust me. So we have that going on. We have things to sort of protect us from the external environment in a way that our species didn't have a few millennia ago, right? And so, Charlie, if you go back a little bit and you're thinking like people just like running around in the grasslands of Africa without any clothes on, parasites crawling on your body is something really important, right? And so if you're feeling things crawling on your body and they're going to give you some disease, then you really need to be able to notify yourself of that quickly and get that off your skin. So often you'll feel something crawling or scratching that it's not even there. You're looking like there's nothing on my arm. But false alarm, it's okay. Better to have a false alarm than no alarm. And there's like a giant tick, like, you know, sucking you dry and giving you Lyme disease. So there you go. That's the cool thing about tickle and itch.
Is that why a lot of people hate being tickled?
I think so, right?
Yeah.
Because there is some level of being tickled that it's like I'm being attacked by an army of ticks. So there you go.
How ticklish are other mammals in comparison to those?
85%.
Good answer.
And that's why he's a professor.
I don't know.
Well, like if
That's an awkward question.
How ticklish are other mammals?
We can't really measure
That's a
that,
thesis.
can we?
Well,
We just
just
can't
go.
really tell if it's affecting them.
No one, I gotta say, Chandler, right?
Yeah.
No one in the history of the world has ever asked that question.
Let's look into that.
Yeah, so I think what you need to do is just randomly grab a mammal and tickle it. And see what happens.
I really tried silverback. I heard that's a great place to start.
Or ask the question better, probably.
Jump
Yeah.
in the enclosure.
I think, I don't know. I mean, I assume they are ticklish.
Just in comparison to like what that does for us, comparison to like how they survive.
I'm going to assume it's something very similar. I'm going to assume that's going to be something that has been evolutionarily conserved. Anything that's susceptible to, you know, parasites crawling on their bodies should have some sort of
Fur.
itch, tickle kind of thing, right? And so I think that would be reasonable. Eli?
So we react to tickling by laughing because we know that we're being tickled, but they react to tickling by aggression. It's what I've like.
Yeah.
Because I looked up if you could tickle a dog because I like.
I never want to know.
I have
Why
a dog,
not?
I have a dog and like I'll like mess around with the fur in between his toes and.
Oh yeah.
They don't like that.
No, he doesn't.
Because the last place you want a parasite's like your feet, right?
Yeah. So
He's really cute.
that was the answer.
The dog's cute?
The
Yeah.
dog is adorable, yes.
I can confirm that.
He's
as
as good
well.
as
Okay, well, there you go. All right, so free nerve endings. We also have encapsulated nerve endings, right? These actually have something around them, as you can see here. That's for things like pressure and vibration, right? So pressure is something we're putting a little more effort into that, right? And if we didn't have that encapsulation on there, if a fly landed on your hand, it would feel like a donkey was sitting on it, right? And that sounds like a bad thing, right? Because you're not feeling, it's not got that sort of filter there, right? To cause a threshold before you feel it as pressure, right? So that's kind of important. And then we actually have things that are separate special cells. And these are things like audition, gustation, and vision. We'll talk about those structures later. This is a photoreceptor, and it has that cool special shape to absorb light.
What about the people that literally have like no free nerve endings? So like the people that will accidentally step on a nail and like never
Yeah.
know, or literally have a whole knitting needle through the bottom of their foot.
I mean, there are a ton of reasons that could cause that.
Yeah.
It could be something central in the brain structure, it could be something peripheral out in the nerve structure or in the connection between the two. But yeah, there are folks who are less sensitive or not sensitive to pain at all. The problem with those individuals is pain is a great thing. Trust me, it keeps you from doing stupid things twice. It's not going to keep you from doing stupid things the first time, right, Reed? But the second time, it's going to keep you from doing it because you're going to remember the last time I did that, my arm was like this for 6 weeks because I broke it. So I'm probably not going to try that trick again. Although some of you are slow learners. Yep.
How does, when someone gets diabetes and like it goes so far as to like break down the like nerves, I'm guessing, how does that impact?
Yeah,
The
so,
different
and we can talk about diabetes in particular is interesting because it does cause damage to the peripheral nerves, right? And so they're not carrying the same messages in the same ways. That can cause a lot of pain, cause a lot of other problems. There are also issues with the retina in folks who are diabetic, and they can actually have macular degeneration. They can actually lose parts of the retina and lose their vision as well. So it really becomes kind of a serious issue. So if at all possible, avoid diabetes. If you've got diabetes, treat that, right? And like see a real medical professional. Sonny?
How do any of these, if they do, work with like people who have an amputated limb that fill them, like the ghosts?
Yeah. We'll talk about that later. How's that? That's a little bit of a bigger conversation, but that's later. We'll talk about it. All right. We also could classify receptors based on their location. There are exteroceptors. These are things kind of on the outside. I know you don't think about your eye or things being on the outside, but compared to things like interoceptors, which are in your blood vessels and in your guts and muscles, they're definitely external. And then you also have proprioceptors. These guys are in your muscles, your joints, your tendons, things like that. I don't want you to take a lot away from this, but I don't know what this is, but this looks like so much fun, right? We can also classify receptors based on the stimulus type. Mechanoreceptors, they're sensitive to some mechanical stimulation, like pressure, something like that. Thermoreceptors, that's temperature. Nociceptors are pain. Photoreceptors, light. Chemoreceptors, chemicals. For us, we're going to think about taste and olfaction. Those are chemicals. And osmoreceptors, those are sensitive to osmotic pressure. We're not going to have to worry about that in this class. All right, here's a great guy, Johann Mueller. We should talk about him a little bit. Right, Kyle? I mean, he's an important dude.
For the fourth time this week.
Yeah, but he's that important. He's that important, right? And so we should really discuss him. Basically, every time you see something, every time you hear something, every time you touch something, it creates an electrical impulse. And that electrical impulse is called an action potential. We're not going to learn a whole lot about that in this class. But that action potential is the same. It doesn't matter if it starts in your fingers, it starts in your ear, it starts in your eye. It's the exact same electrical signal. Now the question becomes, Eli, how in the world do we know what anything is, right? If it's all just the same kind of fun little, I'm going to try to draw it. It's not horrible. Sort of electrical signal, how do we know what anything is? Well, what's important is where it starts and where it ends, right? And so if it starts in the eye and it ends in the visual cortex, then it's got to be a visual signal. We're going to perceive that as a visual stimulus, right, as a visual experience. If it starts in the ear and ends in auditory cortex, well, then we're perceiving that as a sound, right? That's how that works. This is how, you guys have probably heard of cochlear implants. Also retinal prosthetics, that's another thing they're working on now. This is how this works. Cochlear implant, we'll talk a lot about this later. Basic idea is it collects sound through like a microphone, turns that into an electrical signal, and we zap the cochlea with it, right? Or the auditory nerve, because we're bypassing the cochlea. We zap the auditory nerve in the right place, and then you perceive that frequency of sound, right? It's they're much more elegant than they used to be, and the software is better to translate that sound into an electrical potential in the right place, but we know it's an electrical signal. If we know what that electrical signal is, where it starts and where it ends, we can inject that anywhere we want along that pathway and create that same experience, or something very close to it. Auditory is a little easier because we're only dealing with frequencies and amplitude than if we think about the visual system, so our retinal prosthetics are a little bit behind. But we'll get there someday. All right, here's a neuron. Basic things you need to know about a neuron. It's got kind of receptor areas or receiving areas out here. It's going to send an action potential or that electrical signal down, a thing called an axon, and it's going to contact another cell on its dendrites, and that's the basic process. We don't need to think a whole lot about neuron structure in this class, right? But I want to introduce it briefly to you so you can have some understanding when we talk about nerve cells and neurons and brain cells, what these guys might look like, what they're doing, and how that happens. Any questions about this? We'll skip that. And so here's a basic side view of your brain. Later we are going to talk about specific brain structures. There are two large categories or large groupings of this that we'll talk about. One is the cerebral cortex. That's kind of that wrinkly walnut-looking part of the brain that's on the outer side, outside. And then we'll talk about something called the thalamus, and it's this kind of chunk here in the middle, right? For most sensory systems, you'll collect that information, eye, nose, mouth, ears, skin, wherever. You'll send it into the thalamus, and from the thalamus we'll go to various parts in cortex. That's a pretty basic general setup for every sense. There are some exceptions. We'll talk about what those are, okay, as we go along. But just in the very basic way to think about this, we go from the receptor to thalamus to cortex. It's basically the process. Now, there are particular parts of the thalamus that handle vision and hearing and taste, right? And there are specific parts of cortex, like this little guy back here. This handles vision, and this part kind of wrapping around here handles auditory signals. Okay, so it's separate once you get in cortex, but that's the general process. And we will, again, keep in mind we're going in circles, right? We'll come back and we'll fill in a lot of details here where we need to. Skip that. How do we know things about the sensory systems? Well, one, there are lesion studies where we remove part of the brain and then we figure out whether or not you can still perceive that particular stimulus, right? So if we cut out your visual cortex and you can't see anything, then we go, well, that was visual cortex. We know that. We will typically do that in animals. We don't really do that in people. although there are some fancy ways we can do it, people now, like transcranial magnetic stimulation, right? And you can kind of zap part of somebody's brain and turn it off temporarily. So you guys can try this at home if you have refrigerator magnets and electrical tape. Just tape it to your roommate's head while they're asleep and see if they can walk when they wake up. As I was telling Kyle's class on Tuesday, is there anybody else in that class? I feel like there was someone else who was supposed to be in there. I think somebody did show up. I gave a talk once to the Neuroscience Club about how to make your own homemade brain stimulator with a 9-volt battery and some electrical wires. Yeah, that was a fun one. They're like YouTube videos. You can, and they're not videos I made. They're videos somebody else made. Just want to tell you that. I thought about making one. I was like, I gotta try.
You do you.
I'm thinking about Eli's dog now. Because I don't think Eli's going to let me.
Probably never.
Or on the dog.
Yeah, I was
But somebody's-
do it to me before I would let you do it to him.
Really?
Yeah.
Well, there we go. Participant number 1.
Let's do it.
We'll give it a try. You can kill yourself using those, though, so be careful.
That's fine.
Little disclaimer.
Little disclaimer. It's a little one. Slide that in there.
Asterisk, we can kill you, and asterisk.
And they put that on the website. Clinical studies, this is when somebody comes in and they say, hey, I can't see. And then you do some tests, figure out what's going on, and you look at different parts of their sensory systems that might not be functioning, right? We can also do recordings from brain cells. That's pretty awesome. We do this fancy word called psychophysics, right? This is where we would give you some sort of sensory test, right? And we might ask you like, hey, can you see this light? You go, yeah or no. And then we'll change it a little bit, we'll change it a little bit, we'll change it a little bit, and we'll change it a little bit, right? And every time we'll make it just a little bit dimmer and a little bit dimmer until we find like that threshold, right? What's the dimmest light that you can possibly see?
So everything you do when you go to the eye doctor.
Yeah. Yeah, I mean, that's kind of what they're doing, right? I mean, they're doing stuff like that. Artificial intelligence has really become an important sort of aspect of this now as well. Whenever we can, we're going to try to not, put brain stimulators on living things, right? If we can do it with a computer, we will. We'll figure that out. If not, we'll do something else. And of course, brain imaging. I don't know if any of you guys have had any brain imaging. You wanted to share that. Again, that's personal medical information. But if you've had a CT scan, PET scan, MRI, something like that, we can take a look and we go, hey, or fMRI, right? Your brain got really active in these 3 particular spots when you were listening to Pavarotti or something, right? There you go. There's the internet, look it up. I'm trying to help you guys, right? You really need to become cultured and listen to more than, I don't know what you listen to. It's probably too many standard deviations below my IQ to bother with. That was a good one, wasn't it? That's like anytime you throw a stats joke in. Yeah, actually, I don't know. I was like, have you ever tried to watch the Grammys like I did once? And I was like, I don't know these people. It's like I don't. And then the people I had heard of, I was like, man, they're old now. It's like, why'd they invite them out there?
They're like guest performing.
Yeah, I know, right? Hey, here's a great looking guy. This is Pierre Florenz. He was a French guy. He's the guy who developed basically like scooping out parts of brains and figuring out what they do. So that, and that sounds like a really crude approach, and it is, but we didn't have any other approach before this guy back in the 1800s, and he was like, hey, you flip out a chunk, well that's the part that makes the animal breathe, we figured that out. So don't have to do that one again. We have much more advanced approaches now. Transcranial magnetic stimulation is really an extension of this, right? We're not doing something permanent, we're doing something temporary, but it's really the same idea. A lot of this has to do with precision and how we interpret what someone can or cannot do, or an animal can or cannot do once we remove that. Clinical studies, accident, disease, stroke, right? You guys probably know someone who's had a brain injury, someone who's had a stroke, someone or a head injury, someone who has a neurodegenerative disease, and you realize that they lose certain capabilities, right? Some of those are sensory systems, some of them are not. We can study those cases, we can ask those individuals questions, give them tests, and then we can do brain imaging or post-mortem brain examinations, and we can see which particular brain regions were involved. We can learn a lot that way. A lot of this started with this guy. Anyway, he was a Japanese doctor. and he was, after World War II, it's kind of interesting that this is the guy, right? After World War II, he was examining a lot of Japanese veterans, right? So he was back in Japan, and these veterans were coming to him, and he said, "Oh man, I can't see anything over here. I see everything else. I can't see anything over here in this corner." And somebody else said, "I can't see anything over here. I can't see anything there." And so they had these what we call scotomas, right? And so if you look out into the world and there's a black spot or a spot where you can't see anything, that's called a scotoma. Typically this happens because you've had a head injury or a stroke or something. So what he started doing was mapping the locations of these scotomas in these veterans, and then he started mapping their brain damage. And what he found was if you looked at visual cortex and you mapped where they had damage, it correlated exactly to where they could not see in the visual world. And so he was able to create the first map of the visual world onto the brain. And it was really a fascinating sort of breakthrough.
Are scotomas stationary, or can they?
They are I mean, they're stationary because it's always going to be in the same location, but obviously as you move around, that scotoma is going to move in the world, but not relative to your kind of like 0 point. And if you move your eyes, then obviously it goes up as well. Assuming it's damage to primary visual cortex. Now, Josh, there might be some weird situation where someone could have, in a higher visual cortical area, damage that causes them something funky to happen, but please don't worry these people about that, because they don't need to know that. So there you go. I don't know. That's a major breakthrough in like understanding how visual systems work. All right. Don't worry about that. Don't worry about this. Single unit recordings. So what does that mean? We stick a piece of metal into one of those brain cells, and we measure those electrical signals as they're coming through, right? That'll tell us a lot about that particular cell. Does it get excited by a red dot, or does it get excited by a blue dot? Does it get excited if the dot's moving? Does it get excited by a high-pitched voice or by a low-pitched voice, okay? Don't worry about these things. Brain imaging, we've talked about that. Psychophysics, again, basically what we would ask you here, we would show you some sort of set of things and say pick the one that's different than the other two. So we kind of have something at a different orientation here, we have something at a different orientation here. What you notice over time is you keep getting correct responses. You know, it keeps getting closer and closer to that kind of standard, right? And you have to figure out what the threshold is. That's basically psychophysics. I guess we could watch a movie. Anybody want to watch a movie? We'll see if this works.
It's a popcorn.
No, there's no popcorn.
$5 doesn't play.
I'd get my wallet out if I were you. I really don't know what happened.
Good thing we didn't shake, huh?
Yeah, I don't know. There's usually a play button down here at the bottom. Yeah, we'll keep those for now. Is it telling me it's just a picture? Well, it may just be a picture. I really embedded that as a video file, but that's what I get for shifting things around. Yeah, it's not really going to add much to your life to see that, I promise.
Would add $5 though.
Yeah, well, $5.
I mean, it may have. I'll give it to you in imaginary points.
How about real points?
Those don't exist yet. There you go, Mason. +5. Yeah. Don't worry about that. All right, here's Alan Turing. You guys probably know this guy, right? When I brought this picture up, you were probably expecting Sherlock Holmes, but this is the real Alan Turing. Nobody? Alan Turing, what was the name of that movie?
Before our time.
No, it wasn't. It was just like 3 years ago. Yeah, and it had Benedict Cumberbatch in it.
Oh,
Yeah, right? Who also plays Sherlock Holmes.
Something game.
yeah. No. What is that called? It was like.
The Imitation Game.
Yeah.
Yeah, that's the one.
Yeah, because now they have the Turing test, which is kind of the threshold for whether or not a computer is actually like conscious, right? If it can fool you into thinking it's a real person, it's passed the Turing test. It's like a pioneer for artificial intelligence.
It scares me.
That's a little frightening, right? But whatever.
Seems a little bit.
You're going to die before that happens. So.
Hope so.
I mean, now that the government's like telling Facebook to slow it down, you guys are okay. Up until then, it was a little frightening. So Alan Turing, he had a lot of mathematical models, algorithms, things. He was a really genius when it came to those sort of things, kind of the father of artificial intelligence. There's also this guy, Jeff Hawkins, who's like, hey, guys, I figured out like how to create a brain, like an artificial intelligence brain. This guy, Jeff Hawkins, you guys, this is before your time. You may have heard your grandparents talk about this one, the Palm Pilot.
What?
Yeah, it was basically the precursor to the smartphone, right? It was like this PDA that came out. When was that? I feel like that was like the late 90s maybe?
I mean, I was born in the late
Mid-90s.
90s, but I remember my uncle giving me one. I was like, hey, I'm not using this anymore, you can have it.
Yeah, yeah, and they were cool, right? For like 10 minutes until Apple was like, here's an iPhone. And then it's all kind of like, well, they're useless. But Jeff Hawkins made a fortune in computers, doing that and other things, and then he said, hey, I'm going to start up a brain institute. So he's got like the Redwood Neuroscience Institute, where they just figure stuff out about brains. It was kind of exciting. He was smart. He said, hey, computers first, then brains. Some people say, hey, brains, and then there's no money there. I'm going to be honest with you. But he did computers first and made a good chunk of money. There's this idea about the connectome. What does that mean? Like all your brains are connected up differently because you've got like 1 trillion neurons or whatever, right? Some people think if once we figure out like how all your neurons are connected, we'll know things. I don't know. That seems like a big task, and we're never going to do it. So
Because they're connect to me.
what?
Spells connect to me.
I guess if you added like another T in there.
No, connect to me.
Yeah, connect's not really a thing though, Mason. So.
Just say it fast. It works.
Jeez, you know what I'm going to have to do?
Don't think me about points. Hey, my name's Chad, by the way.
You're even now. Back to 0. So anyway, there's this guy Sebastian Soon who's like, hey, if I know like how your system's connected, then that's. But I'm like, well, you know, so I got 1 trillion neurons, and what if like 3 of them are connected differently? Does that mean I'm that much different than anybody else? Probably not, because there's so many of them, right? Now, if I'm an earthworm and I've only got 300 neurons, and one of those is missing or connected differently, that's probably a big deal, right? But if I've got 1 trillion and one of them goes the wrong way, I don't think that does anything, right? It all averages out. Okay. It's like if you bought 1,000 light bulbs and one of them didn't work, would you get upset? It's like, no, I bought 1,000, right? One of them's not going to work. But if you bought 4 light bulbs and one didn't work, you're pretty upset about that, right? You only have 4 light bulbs. All right, studying the senses. We talked about this stuff, localization of function, brain imaging. Got to be careful, studying the sense single units. We talked about that, psychophysics, artificial intelligence. What are some other things we should think about? Transduction. This is a big process. We'll talk about this many times. Transduction is quite simply taking a stimulus and turning that into an electrical signal that your nervous system can use. That stimulus can be light, it can be sound, it can be touch, it can be smell. Whatever that is, we've eventually got to turn that into one of those electrical signals, right? So that Johann Mueller's happy. His ghost will come and haunt you if that doesn't happen. Trust me. That's why he looks so angry back here.
That's unfortunate if you're blind.
But you still have other senses. He's fine with that. Doesn't have to happen everywhere. Yeah, he's happy. If you've got touch and hearing and other sort of transduction things going on, he'll be happy with that. Is this going to be a good one?
Yeah. Is there a reported case of someone missing like a majority of their senses?
I think if you were, so I'm going to say you're going to die. Because remember those visceral senses?
Yeah.
Yeah, like if those things are gone, you will die. Like if you're not monitoring, like if you don't have the chemoreceptors that are monitoring like your blood pH, you can give yourself acidosis and you're going to die. So I think that person may have existed briefly. But we would never know because they died. How's that?
Very
We might know
tentative one.
because they died.
What?
We might know because they died.
Yeah, but then they may, you just walk up on somebody and you don't know, right? I mean, it's just, if you ever find a dead body, I mean, it's a real mystery to figure out why they died.
How many dead bodies have you found?
Well,
I just want the next words out of your mouth to be, hey kids, want to see a dead body.
are we talking human bodies?
Yeah.
My body.
Yeah. That's,
Probably none, I think. Well, like other Ticklish mammals, yeah. Find those sometimes.
It really is a merry-go-round.
I told you, we're going to keep coming back. Hierarchical processing, basic idea here is when you get into cortex, you've got what's called primary, whatever sense it is, cortex. So primary visual cortex, primary auditory cortex, whatever, right? Fill in the blank there. Then you have what we call association cortex, right? So like secondary visual cortex, you've got other higher visual cortex areas. They got very, very creative. Sunny, they named the first one in visual cortex, they named it V1. The second visual cortical area, they named it V2. I know, right? So hang in there. Then they named one V3.
Wow.
I know. Guess what they did for the fourth one? V4. Then they started sometimes using some letters like TE, TEO, MT, MST, right? And that's just to throw you off. And then they'll jump back in with a V8. I don't know. I don't know what, you know, it wasn't my idea. Or I might have done something else. But we'll figure those things out when we get there. But keep in mind, we're going to, sensory stimulus comes in, we're going to break it down into channels, process those individual channels, and then put things back together later. Okay, so you can have that kind of integrated experience. We already talked about Johann Mueller and specific, doctrine-specific nerve energy. Selectivity, this is kind of interesting. If you have a photoreceptor, guess what? It's not there so you can hear stuff. It's there so you can see stuff. It's only going to be sensitive to a specific stimulus and often only to a specific range of that stimulus. So in the auditory system, we have what we call inner hair cells. We'll talk more about these later. You got a bunch of these guys. Some of them are sensitive. They're all sensitive to sound, so that's already selective. Some of those are selectively tuned for 500 hertz frequency, 700 hertz frequency, 1,000 hertz frequency, right? So even within that selective system, you have cells that are selected for particular ranges of that stimulus, okay? So selectivity is very important. Organization, don't worry about it too much. Plasticity, things can change, right? Your cortex can change a little bit. If you, this does not mean that if you lose your sight, all of a sudden you can hear things better than you did before. That's not true. So don't go around telling people that. Yeah.
I thought plasticity meant like flexible.
It is flexible. So it means things, so like there was a study a few years ago with monkeys, and they had imaged this particular monkey, and then they mapped out where its fingers were on its, you know, like for moving its fingers. The monkey got in a fight and lost a finger. It happens. Monkeys fight. They're aggressive. They did a brain image on the monkey again, like a couple weeks later, and what they noticed was the surrounding fingers, that brain region had started to sort of invade that. And so that brain region that was controlling, let's say, its index finger didn't die. It's just the neighboring fingers kind of spread out to cover that area, right? Doesn't mean it was better with its other fingers, right? They could move any faster because you're still limited by the physical apparatus. This is why if you were to lose an entire sensory modality, even if another sensory modality were to invade that cortical area, you're still limited by the input device, right?
So it's more like giving you enough to fill in that gap, not more than enough to fill in that gap and then make a, like,
Okay, so here's a great example. Anybody own a Honda Civic? There we go. How about that? Okay, so we got two Honda Civics, right? And that's got a Honda Civic transmission, right? Let's say that that's awesome. That works because you got a Honda Civic motor, right? And it will move, and it moves as it should, and it moves really well. Let's say I take out that Honda Civic motor, and I put in a Formula One engine. But I left you with that Honda Civic transmission. Not going to happen, right? It doesn't work because that back end doesn't match it, right? It's not going to get you anywhere, okay? It's not going to work. Doesn't matter what you do in cortex, if you've still got the same eyes you started with, you're only going to be able to see as well as the retina is packed with photoreceptors, right? So you've got to think about all the components in that process. And just amping up one or changing one doesn't mean that you've improved anything else to get you any better sort of sensory experience.
So how does LASIK eye surgery work?
So that's a whole different thing. We're dealing with like the lens and some issues, and we can kind of warp that around. Mm-hmm. That's a slightly different process. We'll talk about that when we get to the eye, and we talk about the structure of the eye. It's going to be pretty exciting. Also, I have a laser in my office if anybody wants.
I'll do a quick fix.
Yeah, right?
I'll be-
Why not?
About that $5.
I'll give it to you while you zap my brain.
I know, Josh, we've just got people signed up. Get their contact info after class.
I'll give you your email.
The other thing I want to talk about briefly is noise. This is important. Okay? When I talk about noise in this class, I don't mean useless sound. Okay? That's not what I mean with noise. I'm talking signal to noise ratios. Okay? So, if I look at a red apple, the signal is that red apple, right? The signal is those cells getting excited by that red apple. Now, there are some other cells that are going to randomly, accidentally get excited, right? Okay, and that happens. There's always some baseline activity, even in your visual system, right? If you have no visual input at all, your cells are going to randomly fire sometimes, right? Okay, in your visual system. It happens. That's called noise. You want those cells right on the threshold of actually sending that electrical signal, because you want to be ready when a signal comes through, right? You don't want to catch somebody in sitting on their couch eating a bag of Doritos, right? It's not the way it works. These people need to be ready, right there, get ready to fire that action potential. Because that happens, occasionally, you'll have a little bit of noise. That's fine, okay? The noise, you don't even notice it. It gets filtered out, right? It's not even there. It's not important. So if I were to, I don't know, let me think of a good example here for like signal to noise ratio. What if I am trying to select people who are excellent basketball players? All of us can be basketball players, right? There is some baseline level of basketball skill that every single person has. I'm assuming you can all do this and this, right? And that's something, okay? But there are some people who can do much more amazing things than those two things. And so everybody else is the noise, and I'm looking for the signal. I'm looking for that one person who can, stand really far back and like throw a ball in a hole, from like, the other stadium, right?
So this is strictly like pertaining to sight?
It doesn't
Or
matter.
anything.
It's going to happen in all sensory systems. You randomly have, it happens all the time, like you're randomly going to have cells in your auditory system that are going to get excited, cells in your gustatory system, but they're not, enough of those aren't going to get excited all at once to tell you, yes, I'm tasting a cheeseburger. One cheeseburger cell might get excited. That's not enough to make you think you're eating a cheeseburger, right? You got to get a bunch of them excited. And they're only going to get excited, typically, when you actually have that stimulus.
How does that affect, I'm not going to say the word because I don't know how to say it, that thing where you can hear callers?
Oh, synesthesia.
Yes.
We will talk about synesthesia much later, once we've kind of figured everything else out. Synesthesia is not, it's not a signal to noise issue. It's a, instead of A going to A prime, A goes to B prime, right? And B goes to A prime. And so things are kind of crossed rather than things getting excited when they show. Does that make sense?
So it's like it's wired wrong.
Yeah, things are not, we'll talk a lot more about that later though, so hang in there. Don't drop the class if you're interested in synesthesia, because that's like the last chapter, so there you go. I put the good stuff at the end. Let's see, sensation, talked about this, talked about transduction. Oh, this is kind of interesting. So here's a human brain, here's a brain kind of flattened from a monkey, a bat, and a star-nosed mole. What's interesting about this is if you think about different species use different sensory systems, or they rely more heavily on different sensory systems, right? And so what you can do is you can take a look at the proportion of the brain that's devoted to a particular visual or a particular sensory system and kind of think about that particular animal. So if you think about a bat, for example, look how big, that's this light blue area, this is the auditory area, and then the visual area is just this little guy right here. Okay? So what does that tell you? Well, bats, because they use echolocation, they rely much more heavily on their auditory system, on hearing, than they do on vision, relative to humans, for example, right? And if you think about our kind of auditory area, it's kind of small here, and actually they've underrepresented the visual area. The visual area should be kind of larger, kind of cover this little area down here. So you think about humans, we're a very visual species, right? If you don't believe me, next time you file your taxes, see if you get a tax break for being blind or if you get one for being deaf. and it's for being blind, right? You don't get a tax break for being deaf. You guys don't file your own taxes, do you? No, you guys are just dodging the IRS. Well, they're not going to get you now. You might have a few more weeks to keep outrunning the government, but I wouldn't plan on that being a long-term solution. I feel like eventually they're going to go back to work. That's what I think.
Maybe.
But not today.
If not, it's going to be Mad Max Beyond Thunderdome, and save up on your, spiked football shoulder pads, because I think that's going to be more helpful to you. If you pay your taxes, right? Because your taxes pay my salary. Because I'm a state employee. I'm still getting paid. I haven't really thought about this. Because, the state of West Virginia, you never know what they'll do. It's a real crapshoot.
Teacher strike.
Teacher strike, that was a thing, yeah. It seemed to be effective. I mean, it really worked. Selectivity, organization, noise, plasticity, we talked about that. Sensation, detectability, don't really need to think about that too much. Adaptation, that's really what your lab was about. Taste adaptation. We'll talk more about taste another day, but it was about how does tasting one thing that's powerful change your perception of a substance later, right? And I'd say most of you, when you drank the water after the lemon or the lime or after the sugar, it was different than regular water. For some of you, it may have been the same, but for many of you, there was some sort of different experience, right? That's what we were going for. What else do we need to think about? Don't worry about qualia. Magnitude, that's going to be a fun math thing for if you guys are really interested in logarithmic scales of whatever, but don't worry about that. That's not so important either. Oh, receptive fields is important. We will talk more about this later. It's going to make a lot more sense when we talk about your skin. The basic idea with receptive fields is each cell, and whether that cell's in your brain or it's out actually in your body somewhere, has a specific part of the world, and that's the world relative to that system for which it's responsible. If it's in your visual system, there's a particular point in the visual field that it's responsible for. If it's a sensory, for your touch, there's a particular small area of your skin that it's responsible for, right? So when you get outside of that receptive field into another receptive field, you're activating a different cell. Cortical magnification. What this means is when you get to cortex, not all parts of the world are equal, right? If you think about vision, what's the most important part of your visual field? It's the part in the middle where you're looking, right? Stuff out in the periphery is not as important. So when you get up into cortex, much more of that is going to be devoted to this small little 2 degrees in the middle than what's out in the periphery. That'll make a lot more sense later, but I want to introduce it now. We already talked about noise. Don't worry about Stevens's power law. Or representations. Okay. Questions about that? No? You guys were full of questions.